Hello students, welcome to my channel Online Biology Tutorials. We have just done with the DNA. Now if you have not watched my DNA video, please do watch it before coming to this one, because in that video I have talked about the details of the nucleotide. So please do watch it out, it will be easy for you to understand. The another nucleic acid which we have to study as a part of this chapter is ribonucleic acid. Now when the word formed long way back, many years back, RNA was the nucleic acid. So RNA is primitive in nature in comparison to DNA. But the instability factor is there with RNA. Means RNA is not stable. That's why over a period of time it was replaced with DNA. DNA is more stable because in its pentose sugar, in its deoxyribose structure, it does not contain oxygen. So let's talk about RNA structure. RNA is also an example of polynucleotidic structure. Now when I say polynucleotidic structure, means it is a polynucleotide. Please do watch the video to have a detailed knowledge on how nucleotides are formed, because here I will be just compiling up in the short. So nucleotide is the monomer unit in case of RNA too. Means to make the structure of RNA, the basic unit we should have is a nucleotide. When many nucleotides are repeated, then polynucleotidic or polynucleotide structure is formed. Another thing is that RNA is single-stranded. Okay, RNA is single-stranded, so it will just be represented by a single strand that runs from 5' to 3' direction. But if you talk about DNA, we have already seen that it was a double-stranded structure, and it was anti-parallel. One strand was 5 to 3' and another was 3 to 5'. Let's see the base, the sugar, and the phosphate components in the RNA. So we are studying RNA, and it serves as a genetic material in most of the viruses. DNA is more prevalent because of its stability. Now base will be of course nitrogenous in nature because nitrogen is found in the structure. Sugar will also be found, and phosphate will also be found in the RNA. Bases again will be of two types. They could either be purines or they could be pyrimidines. Okay, purines or pyrimidines. Here there is a difference in RNA, that purines are same but pyrimidines are different. So adenine and guanine, they both will serve as the bases, whereas if you talk about pyrimidines, it is cytosine and uracil. So this is very important difference, that there is no thymine in the case of RNA. So RNA contains no thymine. Very important, in place of it, which base is present? Uracil. This is the major difference. And sugar in the case of RNA is ribose. And if you want to represent it, it will be represented by a pentose ring. This is C1 position, C2, C3, C4, and of course this will be C5. So at its C2 position, OH group is there. So C2 position, OH or hydroxyl group is there, and this is responsible for the instability of the ribose sugar. And the evolution process. Evolution is a process of gradual transformation of one organism to another. So in evolution, always better and stable things, they progress further. Deoxyribose was more stable. That's why we have DNA, and majority of the organisms around us, they have DNA in their heredity as a heredity material. But RNA was being replaced. RNA is more primitive. DNA is recent in its making. And phosphate group is a common to both DNA and RNA. Okay. So let's move on. Here also the nucleotide will be formed by combination of the glycosidic and the phosphodiester bond. Same rules will be applied here. So if you are going to make a nucleotide, and this is a nucleotide of RNA, so of course the sugar in this case will be a ribose one. Base will be attached to the C1 position of the pentose sugar, and the bond will be glycosidic bond. Okay. And phosphate, if you are showing just one nucleotide, then phosphate will be attached to the C5 position. Okay. And the bond present between them, of course, it will be phosphodiester bond. Generally, questions come on DNA. They don't ask you the detailed structure of RNA because they know that there is no point of repetition. So I must appreciate that you should understand each point of DNA very well. So DNA is a polynucleotidic structure. RNA so is a polynucleotidic structure. RNA contains ribose sugar, DNA contains deoxyribose sugar. Bases in case of DNA, they are adenine, guanine, cytosine and thymine. Whereas in the case of RNA, they are adenine, guanine, cytosine and uracil. There is no thymine found. Okay. So when you are going to make the polynucleotidic structure of RNA, it will be similar to what I have explained in DNA. But in the case of RNA, only one strand will be there because RNA is single stranded. So we will not talk about the other strand. So one strand will run from 5 to 3 prime direction. Now let's talk about the base pairing rule. Base pairing rule or complementary base pairing rule. Complementary base pairing rule in the case of RNA. So purine will always bind to the pyrimidine. This is the law. So let's take an example. A will always bind to U because there is no T. Okay. And U will always bind to A. This is one change from DNA because there is no thymine found in the case of RNA. So adenine, which falls in the category of purine, will always bind to a pyrimidine, that is uracil. Here U is pyrimidine and adenine is purine. Okay. The other things will remain common, like C will always bind to G by 3 hydrogen bonds. G will bind to C by 3 hydrogen bonds. Here in the case of AU and UA, the number of hydrogen bonds will be similar to what it was present in adenine and thymine. Okay. So we have understood RNA stands for ribonucleic acid. It is a hereditary material in the viruses. It is more primitive in nature. It is not there in majority of the organisms because of its instability. The presence of hydroxyl group at C2 is responsible for the instability of the ribose sugar. Right? This is important, and the base pairing rule is also important. DNA was double-stranded. RNA is single-stranded. So we will be just writing it from 5 to 3 prime. I hope you have understood it. In the upcoming video, we'll talk about nucleotides and nucleosides. So stay connected and don't forget to share my channel with your family and friends. Thank you.